It started record. Yeah. Okay. So
Thank you, ma'am.
no, thank you to you actually. So yeah. So what I was saying that for genomic library you don't have that option because you will have your clone sequence as introns and exons, even though the whole gene is there. But because in prokaryotes the splicing cannot take place, so you cannot clone your genomic library in prokaryotic system if your final aim is to get the protein out. Then also because cDNAs generally their size is much smaller as compared to the genome. So you don't need to clone cDNA library in BAC or YAC clones. It's not required. So you can clone your cDNA library either in plasmid, phagmid, or lambda or phagmid library. But in this case, it's preferable, at least for organisms which have a very big genome, that you clone as part of BAC or YAC vectors. So these are the general differences between genomic and cDNA library. So in some entrance exams, it asks that what will the cDNA library not have? So you have to means it won't have introns, it won't have promoters, it won't have non-coding region, etc. Anyway, so this library preparation, I have also gone through it 1-2 times, and you have a separate class on that. But for cDNA library, I'll just briefly tell you. So here, what you do is that you take mRNA, or you can take total RNA. There's no need, means specific need that you definitely have to isolate mRNA first, and then only you can make the cDNA library. You can take total RNA, and generally the aim of cDNA library is representation of the expressed proteins. So you generally go for mRNA, and if you want to go for mRNA represented library, then you use these oligo DT primers. Why? Because these mRNAs, they have poly A tails, and because they have poly A tails, so we have an option that as a reverse primer we can use oligo DT. So once you have oligo DT bound to the end of the transcript, then you can use reverse transcriptase. What we will have? We will have a complementary DNA against that RNA. And now you treat it with RNase H. So if you remember, H is the one which degrades RNA as part of RNA DNA complex. So once you treat it with RNase H, then you have these, some RNA fragments are left here. So these may act as primers, and you can, once you add DNA polymerase 1. So if you remember, this again has this 5' to 3' polymerase activity, and also it has 5' to 3' exonuclease activity. So it will remove these RNA primer in front, and then cover up the distance using polymerase activity. So what we finally get is a double-stranded DNA. Now this double-stranded cDNA that you get, you want to clone it in a vector, and you want to generate a library. So what you generally do is you add adapters on both the sides with a restriction enzyme site which is compatible to the enzyme sites which are present in the vector. So you after ligation of the adapters, you cut it with the restriction enzymes, and then you clone it in the vector. So now we have a whole collection of cDNA library, and when you plate it, if it is a phage based library, then you can see it in the form of plaques. And if it is a plasmid library and you transform it in bacteria, then you can see the colonies once you plate it on the plate. So this is the general scheme for cDNA library. And for genomic library, I've already told you that you can use sonication or enzymes. You generate small size, and once you have these fragments, then you can ligate it into any vector. So of your choice by adding adapters, etc. on either side. Now, main problem is that once you have generated the library, how do you identify your clone of interest? That is a big problem, because when we have whole representation of the organism's genome or even cDNA, then obviously it's a huge amount. So you have so many number of clones which are there as part of library. So how do you screen them is one big problem. Where do you find which is your clone of interest? So one very rarely used thing is direct selection. And why it is rarely used, that is because of the fact that it is purely that you are relying on the fact that it is only applicable to those clones which give some survival advantage to the bacteria. So let's say you are looking for antibiotic resistance gene, particular antibiotic resistance gene. So you can, grow your library in presence of the antibiotic. So we are assuming that all rest genes clones will die, but only the clone which has your gene of interest, because it's providing resistance to that particular antibiotic, that will survive. So that is very rarely means, if you are lucky and your gene is giving some advantage to the bacteria, that it will survive and rest will die. But yes, this one option is there. Also it can be used for auxotrophs. Means if you have your host bacteria which is auxotroph or something, and you have made your library, uh, and you're looking for a clone which codes for that particular amino acid, then you can grow your yeast in presence of that. In absence of that, and only the yeast cell which has taken up your plasmid, which has that gene, that will survive. So that's why I told that it's a very rarely used thing. But yes, there is one option that, or let's say, if your gene that you're looking for, it gives some in presence of some substrate, it gives some color. So then also you can grow these colonies in presence of a particular substrate, and only the colony in which your gene is there, that will, that product will react with the substrate and give you color as part of your screening process. So one option is this, but the most commonly used option is obviously based on nucleic acid hybridization, which is purely due to the fact that you look for complementarity between your gene of interest and the probe that you are using. So you need to have some prior knowledge that what is the sequence of that particular gene, and on basis of that you design a probe. So how do you design a probe? You have already been told to some extent. So basically it is a short stretch of oligonucleotide which is labeled. What label we are talking about? It can be radioactively labeled or it can be fluorescently labeled. How do you label these probes? I'll tell you the various techniques that that are generally used. Okay, so the idea is that if your the genes which are not complementary to your oligonucleotide, they those even if it is, binding to some extent, that complex will be highly unstable, and when you give washes while screening process, it will be washed off, or it will not bind at all to any DNA which is present in rest of the colonies, and it will be completely washed off. So the probe will only bind to the DNA of your gene of interest, and there you can see the signal. So how do you actually go from probe designing to finally identifying your colony, where your gene of interest is there or clone of interest is there? Is also based on, so what you do is that you have these colonies, so you transfer it to a nitrocellulose or nylon membrane. So what we have done, you grow your various plates, your library in various plates. You transfer all of these colonies to a nitrocellulose or nylon membrane. Now you have multiple NC and let's say NC membranes, each of which to each of which your bacteria has attached. Now what you do is you degrade the bacteria, and you treat it with alkali and protease. So what will alkali do? It will denature the DNA which is bound to this nitrocellulose membrane. So for each bacteria, the bacteria is lysed. You treat it with proteins are gone, and the DNA which is there, it is now single-stranded, because if you remember, high temperature or alkaline pH, it denatures DNA. Once your DNA is denatured, now what I want is that my DNA should be very strongly cross-linked or bound to this membrane. So for that, I have two options. If I have a nylon membrane, I will go for 2 hours of UV irradiation, which cross-links the DNA to the membrane. If I'm using a nitrocellulose membrane, then preferably I go for 80 degrees Celsius for 2 hours. Sorry, it was not 2 hours of UV radiation. It was either 80 degree for 2 hours or UV radiation. So then what happens that my DNA, it gets cross-linked to the membrane, means it's very tight. So even if I give several washes and all, it doesn't mean that my DNA will flow off. Now, what I want is that I want to probe that which of these DNA is the one which is of my interest. So I add the labeled probe onto this membranes, and the labeled probe will bind to my gene of interest. There's a possibility that it binds to other DNA with some partial complementarity. But then by giving very stringent washes, I can wash it off. So only the region where my probe is very considerably complementary to my gene of interest will remain bound. And because it was radioactively labeled, I will now expose my various nitrocellulose membranes to X-ray film. And when I develop it, I can see one black spot, and that black spot is the my colony of interest. And I can go back to my previous plate and then isolate DNA from it. And that plasmid is my recombinant plasmid, will have my gene of interest. So this is how I can finally look for my clone of interest by screening the whole library. Now, question is that how many of these plates you should make? You have a whole collection of library. So then you have to please go back to my notes. I had told you one formula by which you can calculate that if you are using this particular vector and you are using, and this is the whole, let's say, genomic content of that particular organism, and you're using this particular vector, then how many clones you have to screen. So based on that, you will plate your library on these plates. So here I've shown these, let's say I'm representing 10 centimeter plates, that Petri plates that you generally use. But for screening purposes, library screening purposes, very big NUNC plates are also available in the market. So people who go for screening purposes, they actually buy these big square NUNC plates and grow their library on that, so that they don't have to handle too many plates, but limited number of plates. Okay, so yeah, you will get some spots in the during screening, and you can go back to your how you get your gene of interest. Now coming to that, how do you label the probe? If you're given a stretch of DNA and you want to label it, then which are the methods? You can go for radioactive labeling, or you can go for non-radioactive labeling. So basically it relies on that if you can somehow incorporate some radioactive nucleotide while some polymerization process. Generally that is the way it is used. So the radioactive nucleotide that is generally used for radioactive labeling purposes, it is DATP and alpha 32p DATP. So there are three different methods that are used. The first one is Nick translation, which relies on the fact that when you're isolating a probe, a particular stretch of DNA to be used as a probe, then during that process, there might be some nicks in that DNA stretch. And if these nicks are there, and if I give DNA polymerase 1 along with this alpha 32p DATP and other nucleotides, then what will happen? DNA polymerase 1 will start polymerizing from the nicks, and then it will fill in the gaps, and also it will remove the, because of its 5 prime to 3 prime activity, it can also remove the various stretches to some extent. So as a result, what will happen, that your this DATP, which is radioactively labeled, it will be incorporated at various portions in the whole stretch of probe, and what you will get is a labeled probe. Second is that if your, it relies on the fact that if your probe has some sticky ends at the end, maybe you have generated it after cutting it with a restriction enzyme and releasing it from a plasmid or whatever. So then if there are sticky ends at the end, then you can use a Clino fragment to fill in the ends. And you can also have, means while filling it, you can add radioactively labeled nucleotide as part of the DNTP mix. Then third is by random priming. So in this, what they have done is that you denature by heating, and what you get are single-stranded DNA. You add random, from the market you can buy random hexamers, and you assume that at various places in this whole single stranded stretch these hexamers will bind. So there will be some gaps in between. So if you add Dreno along with DNTPs, which contains alpha 32p DATP, so again you will get labeled DNA. So these are the three different methods that can be easily used for radioactively labeling a particular stretch of DNA, which you can use as a probe. But at the same time, I want to tell you that when you work with radioactivity, I think I mentioned yesterday also, the same exercise, simple, let's say, plasmid isolation or simple centrifugation, it becomes very, very difficult. You can imagine, let's say, if you are making this particular, any DNA stretch, radioactively labeled, then you are using pipette, you are using tips, you have certain wash solutions, and tubes. So all these four things, there is a chance that there will be radioactivity attached to it. So you have to be very, very cautious. So one thing is you have to work in front of a shield at all times. You have to take your hands from either sides and pipette. Always you have to wear gloves, and every tip that you are using has to be separately put in a plastic container, and finally discarded in a separate discard, which is radioactive discard. Every liquid, every wash that you are giving, that particular liquid also has to be collected in a separate beaker or a bottle, and that radioactive liquid waste is also, has to be discarded separately. So every step, you cannot use the same pipettes for other purposes. They should not be commonly used by other lab people in your, other lab people, because they should be aware that this has been used with radioactivity. So they are to be kept separate and all. So every small thing, simple, I would say that simple exercise that you do in like 10 minutes, it will take half an hour or 1 hour. And that also, there is a chance that you get exposed to radioactivity. So in my PhD, I made CDN and genomic library and did all, my whole PhD was on library screening, and that time I used radioactivity. So I know that how difficult it was. But anyway, so now there are various methods where you can use non-radioactive labeling. So here, two examples are given. So in one, how can you make this particular DNA stretch as labeled? So what you do is that as part of nick translation, like I told in my previous slide, you do in presence of biotin DUTP. So this it means in it means AU, in front of A in the template strand, this DUTP will get incorporated. So because it is biotin labeled, so what you get is a probe which is biotin labeled. And how will you detect this biotin labeled probe is by a biotin evident binding. We are very well aware, I think many examples have been told in the past in which I've told that biotin and avidin, these two proteins have a very, very strong binding. So what I do is that to detect wherever my biotin is there, I add avidin molecules which are labeled to a fluorescent marker. So as a result, wherever biotin is there, I will see a fluorescent signal. So this is a simple technique that can be used. Another method is that you use single-stranded DNA, and what you do on the side is that you covalently labeled your HRP. It's a enzyme, horseradish peroxidase, and you covalently label it with polyethylene ME. Then you conjugate, you covalently attach this HRP PEI complex to the DNA probe using glutalaldehyde. So it covalently links the HRP, which is bound to PEI, to these probes, to this DNA stretch. Okay. Now it means that somehow I have on my DNA stretch a particular enzyme, which, when given a particular substrate, that can be a chromogenic substrate, which will give a particular color in presence of that substrate, or it will be a chemiluminescent reaction. So what so it means I have both the options. There is one compound, I forgot the name, which is added, chloro something, which in presence of HRP is converted to a purple color. So it means that wherever my HRP is bound, I can see a purple color when that substrate is given. Or another option is that I add luminol in my while I'm screening, and this luminol is oxidized and gives rise to a chemiluminescence. So that can also be detected by a machine. So what I can do is what these are two different ways by which you can label your DNA probe, either through biotin DUTP and utilizing the biotin evident binding, which has a fluorescent tag, or using HRP, means binding a enzyme to a DNA stretch, and that enzyme in presence of various substrates can give a signal. It can be a colored signal or it can be a luminescent signal. So after coming to that, how do you label the probes? One question is also that what probe sequence, how will you get the probe sequence? So one thing is that if you know exactly that which gene you are looking for, that gene has already been sequenced either as part of the whole genome sequencing of that particular organism, or maybe somebody was working on that gene, so you know the whole gene sequence. So then you can easily design a probe from any part of that gene which can work as a probe for library screening. Another is that maybe many times you're working on an organism in which the genome is not sequenced, you don't know the gene sequence, but maybe some person is very, very interested in that particular gene, and he's working on a protein, and let's say some part of the protein sequence has already been worked out. So in that case, if you know the protein sequence, but you are totally unaware of the gene sequence, what you can do is you can deduce the gene sequence from the protein sequence. How you can deduce is from the fact that we know that every amino acid is coded by certain codons. In many cases, it is coded by a single, not many cases, in just two cases, we know that it is coded by a single codon, while for other cases, we know that one amino acid is coded by multiple codons. So what I do is that I smartly in my protein sequence look for a particular stretch in which the permutation combination of the codons are comparatively less. For example, I have this particular stretch which is marked in red, And the first is TRP, tryptophan, and we know that tryptophan is coded by TGG. There are no other, so it means you have to remember your vowel hypothesis and so on, that every amino acid can be coded by more than one codons. But for tryptophan, we know it is TGG. Similarly, for methionine, we are sure that it is ATG. And for the rest, you will have particular combinations. Like in case of aspartate acid, we know that it is coded by GAT or GAC. So I have these two options. For this also, I have these two options and so on. So what I can do is that I can design my oligonucleotides and then do the screening using that. So basically, it is coming back to that from protein sequence, you deduce the nucleic acid sequence. Not always you can be lucky. There will be many times you will not find any tryptophan here, and maybe there is only first methionine here, and in between the protein there is no methionine. Then you have no option, but you have to make different combinations of oligonucleotides and try it out whether they can, because we know that even partial complementarity can lead to binding of the oligonucleotide to its target gene sequence. So we can rely on that fact also, and identify the clone which has my gene of interest. Then comes third option. There is a possibility that I don't know the protein sequence. I don't know the gene sequence for my gene of interest. I don't have means no idea. But I know that, for example, I'm interested in cytochrome C, and my aim is to know whether neurospora has cytochrome C or not. And if it has, then I want to know its gene sequence and I want to isolate its gene. So what I do is that I know that in a related species, let's say in a yeast, I have this cytochrome C gene sequence, which I completely know. So we know that many species, these gene sequences, they are conserved. At least some part of it is conserved. So based on my knowledge that, okay, generally these part of this particular gene is shown to be conserved in various organisms, I can design a probe from that particular stretch from a heterologous, I mean from a different species, different organism, and then do the probing. So what because we know that the probe, even if it is partially complementary, and if I can regulate my stringency conditions while washing, it will bind to that particular gene stretch. I'm assuming that I can fish out my gene of interest. So this is how it is done. And similarly for but means only thing is that you have to be intelligent enough that if you want to look for, let's say, a gene sequence in neurospora, then preferably you will take a gene from a fungi or from yeast, but you will not preferably take the gene sequence or the probe from, let's say, a plant or a human, where there are chances that there can be more divergence. So we will look for related species. If you are looking for to get a gene sequence from a monocot, then I will preferably take a heterologous probe from a monocot and not a dicot. So these things you have to keep in mind when you are going for heterologous probing. Similarly, if this example I've told you before also, if you are looking for a whole gene family, then what you do is that you regulate the stringency condition. So let's say for a particular gene, a gene family consists of A to E. Let's say phytochrome A, phytochrome B, C, D, E. Let's say all these. But I just have the gene probe sequence of phytochrome A. So what I can do is I can use that probe sequence to screen the whole library, but I will use comparatively lower stringency, that even if some partial complementarity is there, then it binds to it. So then I can find the various gene family members. So multi-gene family, I'm just I think you might already be knowing, but it means that those members in the family, they all have quite good amount of complementarity, or let's say sequence similarity in their sequences, and many of them may also have similar functions. So it means that we are relying on the fact that the gene family, many of these will definitely have some sequences which are common between these. So based on that, you can also look for the gene family by heterologous probing. Now, one point is that, see, when you have to take a probe, one option is that you, let's say, uh, means cut it from a particular gene fragment and then get the gene probe, or you have to, in many cases you will have to synthesize it. For example, when you send your primer synthesis, so how it is done, so how do you synthesize from scratch a oligonucleotide where you don't have a template? Otherwise, in many cases, you have a template, and then you, using Clino fragment or whatever fragment, you can synthesize your probe, which is complementary to it. But what about those cases where you have to start from the scratch de novo and you want to synthesize it? So, what method is used? So, this is called as chemical method of oligonucleotide synthesis. So, let's see how it is done. So the in TA Brown book, only this figure is shown and only this legend is shown. So more details are there for you in the notes section, but briefly I will tell you. So what you do is that in this case, you take the nucleotides, both ends, the ends of which have been either protected or activated. So in general, you take nucleotides, the 3' end of which is activated and the 5' end of which is protected. Okay. So what will happen that, and you take some polymeric substrate, in this case, they have taken a silica support, and you attach the first nucleotide to it. So what will happen that T will bind to this activated 3' end, binds to this silica support. And on the 5' end, we will not have multiple T's bound, which we don't want, because I want to synthesize a oligonucleotide of a particular sequence that I'm aware of. So in that case, I don't want more T's to be added at one go. So I ensure it by protecting this end. So what will I do? How do I add the next nucleotide then? So for that, you have to remove the protection. Once it is added, then you remove the protection and then you add the new nucleotide. So this new nucleotide, because it has a activated 3' end, it through condensation reaction, phosphodiester bond will be made and G will be added to it. But it will ensure that only one G is added to it because this end, 5' end, is protected. So again, you remove the 5' end, the protection, and you add the third nucleotide. So in this case, it doesn't mean that, let's say, my final sequence I want is TGG ACT, whatever. So I know that I will have two Gs in succession, but at the same time, because it is a regulated process, at one time only one G will be added here. So then I will add the third nucleotide, again the G, and then again the condensation reaction will take place, and this thing will be formed. So this is how your oligonucleotides, they are synthesized in a particular company by this particular method. And once it is synthesized, it is taken off from the silica support and the protections are removed. And then these oligonucleotides, they are either page purified or they are HPLC purified and then given to the user. So generally for normal PCR reactions, for small these oligos, you generally opt for page purified, but otherwise, if they are very big in size, then you generally go for HPLC purification and get the primers or probes for your gene of interest. Okay, so let's say that you finally identify and you get a clone in which you got the signal, and you know that, okay, your gene of interest is surely present in this clone. But you might be knowing that BAC, yak clones, or even these phages, etc. They have very big insert size. So from this big insert, where how exactly do you find your gene of interest? Okay, so because the inserts for a particular recombinant clone, they will be very big, and your gene will be just a small part of it. So how do you actually identify that where your gene is there? So you can go for southern hybridization and then actually going back to the particular fragment where your gene of interest is there. So in this case, what they have done is that take your clone, you cut it with a particular restriction enzyme. You generally go for a 6-base pair cutter, not a 8-base pair cutter, because then you will have very less number of enzyme sites in there, and again the fragments will be much bigger in size once you cut it with the 8-base pair cutter. So here you go for a 6-base pair cutter, and then you get these various fragments. These fragments, you run it on a gel. Now among these fragments, I want to look that where is my gene of interest. So what I do is I again go for hybridization. So for hybridization, every time you will require a probe. So again, you will generate a probe which is specific to your gene and which is radioactively labeled or fluorescently labeled. So now, how do I probe it on a gel, which is very difficult? So obviously, I will have to transfer my these fragments to a membrane. So how do I do that? So this is the setup that which is generally done for southern hybridization, and it is called southern hybridization because the scientist called E.M. Southern, he devised this technique. So when you are doing screening for DNA fragments, you say it's a southern hybridization. Same screening for RNA fragments, then you say it's a northern hybridization. And that's why when you screen for proteins, then you say it's a western blotting. So this is southern blotting technique when DNA is involved. When you have to transfer a RNA fragment to this membrane, then you say it's a northern blotting. And if you transfer the proteins to a membrane and then screen it, it's called western blotting. Okay. So anyway, so how do you transfer the DNA to membrane? So what you do is that you place your gel, this blue line, on a particular support, and then you underneath the gel there is a wick which goes from here to here. What is this wick made of? Generally, you use a 3 mm Wattman paper. So it's a thick wick, I mean broad wick, which goes from here, and it is underneath the whole of the gel, the whole width of the gel and the length of the gel, and then it goes here and it is soaked in a buffer. And the whole idea is that through the capillary action, the DNA fragments move from here, from the gel, to a particular paper, in this case, nitrocellulose membrane or a nylon membrane. So, what we have a wick, then a gel, then a membrane, which is in direct contact with the gel, and above the membrane you have a whole set of paper towels. I mean, you can use any sort of blotting sheets, a whole stack of it, and it is believed that with the capillary action the DNA will be moved from the gel to the membrane. And because the pores of the membrane are much smaller, so it doesn't go from the membrane to these paper towels, so it gets attached here. So this is how you can transfer your DNA to a membrane, and then you go for hybridization, which is exactly how you did for previous thing. You dip your nitrocellulose membrane or nylon membrane. First, you will fix your DNA fragments either by 80° for 2 hours or for UV irradiation. And after fixing, you will add your probe in a solution. It will bind at various places depending on how stringent your conditions is, and obviously it will bind to your gene of interest as well. And through washes you will remove some of the binding which was not tight or not strong enough, and then you are left with your probe binding to only your gene of interest, and how you will detect it by exposing it to the X-ray film, and you will get a signal. Once I get the signal, I will go back to my gel, and I will know through means I would have already taken a photo of my gel, and I will compare that this signal coincides to which particular fragment. Once I get that fragment, then I will cut it out from the gel, and then I can clone it further, or I can directly send it for sequencing to get my gene sequence, and that's how I will get my gene of interest. Okay, so one last method of screening is immunoscreening, and it is obviously only applicable to those screening of the library where the protein products are being made. And because immunoscreening, it is based on antigen and antibody reaction. So obviously, it is also limited to only those proteins for which we have antibodies available. If you don't have the antibodies, then you cannot go for immunoscreening. And yeah, so means it's very limited option. But yes, there is a possibility if you are looking for a particular you know the product of a particular gene, and you already have an antibody against it, then to identify the original gene clone and fish it out from the genomic library, you can go for immunoscreening. So as I told you, it is based on antigen antibody binding. So let's say if you have your protein of interest and you want to generate a specific antibody against it, if it's not already available in the market, then what you will do is you will inject this whole protein, or maybe a small epitope of it, or let's say a small part of it, in various these animals. Generally, mice is used, or rabbit is used. In some cases, goat is used, or sheep is used. These are the general animals in which, for commercial purposes, also these antibodies are purified, generated and then purified. So you inject your protein, whole protein or a part of protein, specific epitope into the bloodstream of these animals, and it is the idea is that the animal will identify these as foreign. If it identifies these as foreign, it will generate antibody against against it, and then you harvest the antibody which is generated against that protein from the blood of these animals. So this is how you get purified antibody against your protein of interest. So how is generally immunoscreening done? You take the membrane, you take the colonies like you did previously. You transfer those colonies to a membrane. You lyse the cells, and then you add your specific antibody. So your specific antibody will only bind to those places where your protein of interest is there, and not to rest of the places. So once your antibody binds to it, how do you detect it? So there are three different methods. One option is that the antibody that you are using, maybe that itself has a label. It can be a radioactive label, or it can be a fluorescent label. So there are some antibodies which are already available from the market, which are fluorescently labeled. Another option is, which is generally preferred, is that you recognize these primary antibodies by using protein A, which is radioactively or fluorescently labeled. So what is protein A? So protein A is a protein which recognizes the constant region of various antibodies. So wherever your antibody will be there, protein A goes and recognizes it based on its affinity to its constant region. And because protein A itself is labeled, I told you it can be radioactive or fluorescent, you can detect the signal. Third option is that you have your primary antibodies bound to it. You get from the market the secondary antibodies, which are fluorescently labeled from the market. or radioactively labeled. Generally, they are fluorescently labeled. And then you add on to it. So, what are secondary antibodies? Again, secondary antibodies are the antibodies which recognize these antibodies. General antibodies, mostly it is the constant region. So, these secondary antibodies will go and bind to it, and because they themselves have a label, you can again detect it on a plate. So, or a membrane. So this is how immunoscreening is done. Everything is same except the fact that instead of DNA interactions, or let's say DNA RNA interactions, what you are going for are antigen antibody interactions. And you obviously cannot screen for the genes or the regions which are not coding for a protein. So that's all for today's class. So what we have learned is I've told you what are the general differences between genomic and cDNA library. And I've told you various methods of screening, where I've told you can go for direct clone selection, which is only based on uh, the fact that the gene that you are interested in is giving some advantage for survival to the bacteria, or it is giving some colored signal in presence of a substrate. So generally, this is a very limited approach, only applicable to very rare cases. And generally, you go for nucleic acid hybridization. And I've told you that for nucleic acid hybridization, you have to make a probe. For probe making, you can go for radioactive probe or non-radioactive probe. And also, you can go for oligonucleotide synthesis completely when you don't have any template DNA in hand and you want to get the probe synthesized, then you can get it done by adding one base one by one according to your sequence of interest. Then I've already told you that the probes can work if there is an exact nucleotide match, and if you can fiddle with the this washes and the stringency of washes while you're doing hybridization. You can also do heterologous probing, which is based on the fact that even if probe is not completely matching the template, then also you can see a signal. And I've told you the technique of southern hybridization and also immunoscreening. So, open for questions. Okay, so there are no questions. Okay. Is there some? Please start the recording, ma'am. Okay. So anyway, so then today I can definitely say happy Dashera to all, right? We are not meeting till next week.
Happy Dashara, ma'am.
Okay. Yeah. Bye bye then.
Thank
Happy Dashara, ma'am. Bye.